Hello everyone, welcome to the next episode of Microtalks. Today we will be discussing one of the interesting features of microbial growth, namely synchronous growth. This is not a normal growth pattern that is seen in the bacteria. In fact, synchronous growth is something that has to be experimentally achieved. Synchronous growth is an important aspect with respect to applications in microbial research. Synchronous growth in bacteria refers to a growth pattern in which a population of bacterial cells enters the various stages of the cell cycle at the same time, as opposed to asynchronous growth, where the cells in a population are at different stages of growth and division, like in a normal growth cycle. So synchronous growth therefore is something that is experimentally attained or experimentally achieved. So there are various methods or various techniques with which you could bring about synchronous growth, or there are different techniques, synchronization techniques. One of the most commonly used synchronization technique is nutrient depletion. So nutrient depletion basically is a method that can be employed to synchronize the growth of bacterial populations. Initially, the cells are in a different stage of their life cycle in any population of bacterial cells. And the cells also grow at different rates depending upon the availability of a particular nutrient, and also depending upon the capacity of every individual cell to take up the nutrient, to assimilate that nutrient. When the nutrient is depleted, bacterial cells will halt their growth. So normally the nutrient that is depleted, usually in order to attain synchronicity, is either nitrogen, phosphorus or carbon. So once either of these three nutrients is depleted, the bacterial cells will obviously halt their growth and they will go into a resting mode. Once the nutrient is restored after a period of depletion, all the cells that were halted in the stationary phase due to nutrient depletion will resume their growth at approximately the same time. Thus, there will be cells of around the same age that will enter the exponential phase in a given population. Thus, cells can be harvested at a specific growth phase. The growth rates can be controlled by varying the time as well as the degree of nutrient depletion. The other method that is very often used is filtration. Filtration will remove the cells that have already divided. Filtration will separate the cells based on the size and will help in collecting those cells which are of a similar size. Assuming that the cells that are of a similar size will also be in a specific growth phase. So with this basic assumption, filtration is also commonly used to attain synchronicity. Another method that is commonly used to attain synchronicity is the centrifugation, or more specifically the density gradient centrifugation. This can be used to separate the cells based on the size or based on the density of the cell. Flow cytometry is another commonly used technique which will separate the cells based on their size. So you have an assortment of different synchronization techniques that can be used to collect cells in a specific growth phase or cells of a specific size. How do you monitor synchronous growth? Monitoring the turbidity by measuring optical density obviously is one of the easiest methods that can help us to monitor the synchronous growth. The turbidity can provide data on the cell density. If you look at the optical density, it will give you the certain indication of whether the cells are in the same growth phase. Besides that, more accurate method is live cell imaging, which can be done by fluorescence microscopy. This helps to visualize the cellular processes in a real time. And of course, carrying out viable count of the cells. So measuring the CFU periodically during the growth phase will help to assess the growth and division of the cells. Let us look at the applications of synchronous growth as well as there are the limitations of synchronous growth. The common applications of synchronous growth are seen in the area of microbial research, where you have to study the cell cycle dynamics, genetics. Commonly, synchronous growth is attained when you are carrying out experiments in molecular biology, in biochemistry, and it is important that synchronous growth has to be maintained when carrying out a particular experiment, especially when you are also studying the effect of certain drugs on the growth of bacteria. It has limitations, no doubt. Obviously, it is technically very challenging. It is difficult to get the cells in the same phase of growth. So it requires continuous monitoring. And if you are using nutrient depletion as one of the techniques of attaining synchronicity, then the cells are put under stress. And once the nutrient is given back to the cells in the medium, there will be a considerable lag phase for the cells to overcome the stress to which they had been previously exposed. And last, synchronous growth does not mimic natural growth patterns. So the growth curve that you see in synchronous growth will not be the same S-shaped curve that you would otherwise see in a natural growth pattern. I hope the video clearly explains to you the basics of synchronous growth and you have understood the limitations and applications and how to achieve synchronicity. Thank you everybody for watching this video. Please do like the video, share it and subscribe to my channel. Thanks for watching once again. Looking forward to bringing you more episodes of Micro Talks.